So Carrie and I are going to talk about coming down the pipeline, what's new in CF. And you think, CF, why have we got a foot on our first slide? Well, this is the foot of a mother of a CF patient, and she is having a tattoo done. And the tattoo is the Kalydeco molecule. So that's why we've got it on our slide. Mum's just so excited that Kalydeco is available. She's having it tattooed on her foot. Next one. So disclosures today, Carrie and I have no financial interest in any of the companies involved in this talk, and the below are the investigative team, are the companies that we are involved with our research at the moment. So our research team here at Maine Medical Center in Pediatrics is Dr. Carnes, Mary Ellen Corrigan, Dr. Mello, Carrie, Harmony, if you want to wave, Harmony, she's here, and Olga Diomede, and hopefully I pronounced her last name correctly. And on the adult side, Dr. Zuckerman is here today, Dr. Noft, Tina Manley is here, have a wave, Rhoda Kennedy-Dudevois, and Rebecca Violet. So we have a really good team of researchers here at Maine Medical Center, both in the adult and pediatric side. Next one, please. So just to review the topics that we're going to be going through today, Carrie's going to be talking about how CFTR works. We're going to talk about drug development, new treatments, current projects for research at Maine Medical Center, and upcoming CF research projects that are coming through the pipeline. And Carrie's going to start us off by talking about the CFTR and how it works.
Good morning, everyone. I hope you've all had your coffee today, because we are going to start out with a science lesson. I can see the eyelids drooping already, but we'll try to keep it brief and interesting. And I know that all of you in this audience probably have heard about how the CF gene works over and over again, but I think it's important that we revisit it today because it's going to be useful in helping us understand the new therapies that are coming out, as well as where research is headed in the future. And so I'm just going to do a brief overview of how the CFTR gene works. And so, as you all know, everybody has 2 copies of the CFTR gene, and when they're working properly, they make a protein that's expressed in the epithelial cells of a variety of organs and cause our body to function how they're supposed to function. Now, in patients with CF, we have changes in the 2 CFTR genes that cause problems with the CFTR protein either being made or the way it works, which can cause problems in a variety of organs and bring out the clinical traits we see in cystic fibrosis. If you'll hit the button, and then again. So I just want to go and show you a little bit more detail about how the CFTR gene works. And so you'll see these purple structures, which are the CFTR proteins, and they are housed in the cell membrane, and you can see these little blue dots, which are chloride ions. And what the CFTR protein does is it acts as a channel to move the chloride ions out of the cell and create a nice balance inside and out of the cell. Now, why is this important? And do the next. So here we have shown a normal healthy lungs, and you'll see that the balance of the chloride ions is nice and even in the cell and out of the cell, and this creates a nice thin watery mucus that can easily be moved out of the lungs and get the bacteria away from the lungs. And you'll see in the CF lungs that the chloride ions are not being transported outside of the cell, which creates a thick, sticky mucus, and that traps the bacteria inside and doesn't allow it to get moved through the lungs and out of the body. And so I just wanted to show you what it really looks like in a human epithelial airway cell. And you can see highlighted in bright blue is the nucleus of the cell, which houses all the genetic material that tells the cell how to function. And then we have the goblet cell, which produces mucin, which when combined with water makes the mucus that we trap the bacteria with to keep our lungs clean. And in the red is the cell membrane where the CFTR gene or protein is housed. And so that's where the chloride ions need to be transported out of to create the mucus that the cilia then move away. So if you indulge me for one more moment, I'm going to show you how it actually works. Okay, so here we can see we're going to zoom in on the cell membrane. And there's the cilia beating away. And this is nice, thin, normal mucus coming through. And you can see how the cilia are just pushing it through very easily. There's no problems here. And then we're going to zoom down to the cell membrane, and we'll see what's happening with these CFTR channels. And there they are, just pumping away. Everything's working great. And now you start to notice that something's, slowing down. The chloride ions are not getting through. And this is what's happening in CF. We can't get the chloride ions above the surface of the cell. And this is creating a thick, sticky mucus that you're going to see come down. And the cilia are trying to get that thick mucus out. And it can't happen. They get weighed down. And so this is why bacteria will stay in the lungs in patients with CF, and we can't clear it out as easily. So now that we know how CFTR works, we can apply that information when thinking about future therapies. With CF, there are many different paths that we can take when thinking about research to do. And so what would be great is if we could correct the underlying gene mutation, and then all the symptoms of CF are corrected. And so there are gene therapy studies that are going on working on this. But with gene therapy, we've known from other studies in other diseases that it can come along with a lot of complications. And so these studies tend to move a little bit more slowly down the drug pipeline because there's a lot more testing that has to be done to make sure it's effective, short-term and long-term, as well as safe for the patients. And so these may not make it through the drug pipeline as quickly as some of the other treatments that we have. Now there's also the problem that different CFTR mutations they all work in different ways. And so one treatment is not ideal for every type of CFTR mutation when we're thinking about correcting the underlying gene mutation. And we're going to talk about that a little bit more later in this talk. So there's some mutations that cause abnormal CFTR protein. There's not enough of it. And then that's what's causing the problem getting the chloride ions to the surface. Or there's also mutations that cause altered ion transport. So the CFTR protein is present, but for some reason, you can't transport things through it. And so that's what we call potentiation. And so we need to restore proper ion transport in those mutations. And then we also have studies that are coming that look at treating the symptoms of CF, infection control, infection in the lungs, inflammation in the lungs, and other organs. And so all of these lines of research are very important, and we need them all so we can have our patients to stay healthy, so that when the underlying gene can be treated, our patients are there and ready for it. And so I'm going to turn this over to Karen to talk to you about how we come from the idea of treating the disorder to clinical treatments down the pipeline.
So Carrie talked about the drug development pipeline, and this is what it looks like. So drug development and drug discovery is a very long process. It can take 10 to 15 years. And to start with, at the very beginning, at drug discovery, the scientists are working in the lab, looking at compounds, and if these compounds look promising, they go on to animal testing. And once they've completed animal testing, then we can move on to phase 1 clinical trials, if you could push the button. And the phase 1 clinical trials are looking at human safety. Normally these trials are done in healthy volunteers, 20 to 100 or so people. They last a couple of months, looking at the safety of a particular drug or compound. And if it looks promising and things are working, and about 70% of drugs that go through phase 1 are able to go on to phase 2. Click the button. And then we're really looking at studying the drug in a particular patient population, in what we're talking about, cystic fibrosis. This is usually done at multiple centers, 100 to thousands of patients. The studies are looking at safety, they're looking at efficacy, and by the end of phase 2 trials, about 30% of these drugs are successful and able to go on to phase 3 trials. And phase 3 trials are the definitive trials. They're usually large national trials, often international. These are the trials that we're mainly involved in here at Maine Medical Center. They are, again, looking at safety, at efficacy, the benefits, side effects profiles. All this information is gathered, and at the end of the trial, the drug company puts together all the information, analyzes all the data, and presents it to the FDA. And the FDA reviews everything, goes to and fro with the drug company, and then at the end of the day, hopefully the drug is approved, and that's when it's available to patients in the market. Phase 4 are post-market studies. So patients are on the drug, but post-market studies are looking at continued safety profiles, looking at side effect profiles and the benefits. So there are many stages to research, and as I said, it takes 10 to 15 years to get a drug to market, so it takes a long time. And these are some of the drugs that are in the CF Foundation's therapeutic pipeline. Along the bottom, you'll see the various phases of trials, and down the side, you have the categories that the trials are in. You can see we've got several drugs that are now available to patients, and a lot of drugs in phase 3 and phase 2 trials right now, some of which we're involved with here at Maine Medical Center. So what therapies are we looking at having here in the near future? So one of the studies we were involved with here at Maine Medical Center was the long-term inhaled dry powder mannitol study in cystic fibrosis. And mannitol restores airway surface liquid. It's an osmotic agent. It's actually a sugar. And what it does is it draws water out of the tissues and into the lung. When you inhale the mannitol, it draws that fluid out into the lung. It hydrates the mucus so it clears more easily, and the cilia can work. If you remember the video, you saw how flat the cilia were and the mucus wasn't moving. By pulling that fluid in, it thins everything, the cilia can start wafting again, and the mucus is cleared. This study has completed its phase 3, and we do have some results. So in this study, it was a 52-week study. It was a blinded study for the first 26 weeks. So some of the patients received mannitol, some of the patients received a placebo for the first 26 weeks. The blue line shows the patients that received mannitol. Down the bottom is baseline, and this shows the mean change in FEV1 for those patients on the blue line that were taking mannitol. And as you can see, there was a lovely increase in mean FEV1. The red line is the placebo. So those that were taking placebo, you can see there's very little change in their mean FEV1. At 26 weeks, all the patients that were on the placebo were entered into the open label extension and were able to receive mannitol as well. And you can see on the second 26 weeks, the blue line, patients that were already on the mannitol, their lung function was maintained. And the patients that were on placebo but then got mannitol, their lung function came up to the level of those that had been taking mannitol the whole time. So it's a very encouraging study.
I want to explain what FEV1 is.
FEV1 is lung function. I'm sorry. So just to review the study, the treated group had a mean improvement in FEV1, lung function, of 105 mLs, which is 8.2% above baseline. The treated group had a relative improvement in FEV1 of 3.75% versus the control group or the placebo group. Adverse events and sputum micrology were similar in both treated groups. Exacerbations rates were low in both groups, but was not statistically significant between both. In the open label extension study, lung function was maintained in the original treatment group, as I've said, and the control group's lung function came up to that level of the treated group. and there were no qualitative changes in microbiology results from baseline in either group. So why are drug companies looking at inhaled therapies? Why are we using them? Why are you being asked to use more and more inhaled therapies? And if you think about the location of the disease is in the lung, it's endobronchial, it's in the lung. And aerosol delivery results in high drug concentration in the lung where the disease is. It results in low systemic or in the bloodstream levels, which improves safety, and so it optimizes safety and effectiveness. What inhaled therapies are available now? We have bronchodilators like albuterol, anti-inflammatories, we have Dornase, hypertonic saline, and the antimicrobials Toby, Caistin, and colistin. Coming through the pipeline in the future, we have mannitol that we've just talked about, ASL modulators or airway surface liquid modulators, antiproteases which target inflammation in CF, and glutathiones which are antioxidants, and new antimicrobials, Cipro and levofloxacin, we're being studied right now. So with these new therapies, we're looking at adherence. How can we improve adherence and reduce the time burden for patients? Because time is a huge issue for most CF patients. And when we look at refills of tobramycin, inhaled Toby, the percent of patients that are refilling their Toby prescriptions twice or less a year is about 71%. Patients refilling 2 to 4 times a year is about 22%, and only about 6% of patients are refilling their Toby more than 4 times a year. Why is it important to use your Toby and be adherent to therapy? If you look on the right side, the red bar shows that those that are admitted to hospital more often are those that don't fill their Toby more than twice a year. So you're much, much more likely to be admitted to hospital if you're not adherent with your Toby cycles or any other medication cycles. So what can we do to improve adherence? So not only are the drug companies looking at new medications, they're looking at new delivery systems to reduce that time burden. So with Toby, TIS, you're using a jet nebulizer, PARI jet neb. You do it twice a day. Prep time's short, it's 1 minute. But total delivery time and total cleaning for the whole day adds up to about 56 minutes, close to an hour. Then Caistin, as in the Altera came along. That's 3 times a day. But overall, we're reducing the time burden by almost a half to 32 minutes. So we're getting somewhere. We're working on reducing that time burden even more. Now we have dry powdered inhalers coming along. Mannitol. Tip is tobramycin. Look at this, down to 10 minutes a day. So it's going to be much easier for patients to take their medications. So what is the optimal aerosol delivery device? Most desirable, most favorable. It needs to get the most drug in the lung, in the right place in the lung. It needs to be fast. It needs to be easy to clean up. It needs to be simple to use, inexpensive, ergonomic, and for the teens, it needs to be cool. It's got to be purple for the girls or something. And that picture with the smoking man climbing up, I deny all knowledge of that. I shamelessly stole this slide from somebody else. So dry powdered inhalers, they sound really exciting. So what are the pros and cons? They're small, they're portable, no power source, they can go anywhere. They're capable of high payloads, you can get a lot of drug in a quick period of time, but they do require adequate inspiratory flow and volume. It can increase cough, and we can't use them in children under the age of 5 or 6. So looking at some of the other studies that have been done and other medications that are available, we've seen that mannitol increases lung function, but what do the other drugs do? So hypertonic saline in studies showed to increase lung function by a mean of about 5%. Toby in the second bar, it's about 10 or 11%. Pormazyme, about 8%. Azithromycin, about 6%. increase in lung function, and inhaled Caiston, again, about 8% mean increase in lung function. So we can see that by using these inhaled medications, we really can improve lung function. Carrie is going to talk about a medication that has just been approved and other medications that are coming down. Right now, it's very exciting time in CF, but the emphasis really has to be on maintaining lung function, being as healthy as you possibly can, so these new medications coming down the pipeline are going to work the best. In conjunction with looking after your health and keeping as healthy as possible and keeping your lung function as high as possible, you need to know your genotype. Most of patients here know them, but there are patients that have not been genotyped or have one unknown genotype. And the CF Foundation have a program called the Mutation Analysis Program that provides free genotyping to patients that either have never been genotyped or have one unknown mutation. So if you are in that category, I encourage you to talk to your physicians, get on this program, because together with knowing your genotype and being as healthy as possible, you're going to be able to really benefit from some of the new medications coming through the therapeutic pipeline, and Carrie's going to talk about those right now.
Yes, as Karen said, it's very important to know your genotype, and I think for those of you who have been following the Vertex story, and I imagine that's everybody in this room, we have just had the approval of the first ever CF drug for CF that treats the underlying genetic cause in mutation G551D. And so it's important that people know because their mutation, because more studies like this are coming, and for other mutations. And so we want you to be able to be involved in these studies as they come and the medications as they're approved. And so G551D makes up about 4% of the CF population in the United States. And so the question that's probably on everybody's mind is, why doesn't Kalydeco work for other mutations? And so we're going to talk, I hope you remembered the science lesson that I gave you earlier, because that's going to come in handy as we talk about what Kalydeco does and why it doesn't work in all mutations. So a little bit more cellular biology here. You can see on the left-hand side of the slide the normal CFTR protein channels and the chloride ions. And you can see the chloride ions are pretty evenly distributed, and there's quite a few CFTR proteins on the cell surface. And that allows everything to function properly. In CF mutations, we can have two types of changes that happen. And some can cause problems with the quantity of CFTR protein at the cell surface, and some can cause problems with the function, as we had discussed earlier. And as you can see, delta F508 falls into the first category, where the quantity of CFTR protein is affected. So no matter what we do, we are not going to be able to get the same amount of chloride ion through if we don't have channels available to get it through to the cell surface. And so you can see the G551D falls into the other category, where function of the CFTR protein at the cell surface is affected. And you'll notice that the CFTR protein is present in normal numbers in the G551D. But what the problem is that there's a gate that is closed, and we can't get anything through the gate. And so all the symptoms in the end are the same for delta F508 in patients with CF and other mutations, but the function of how it gets that way is totally different. And so Kalydeco was discovered as the key to this gate in G551D. And Vertex is looking at other mutations and searching for ways to correct those problems as well. And so let's see what Kalydeco does for patients with CF. So here you can see, so Kalydeco is also known as VX770 in its research phase, and ivacaftor. So a drug company can't keep one name throughout the whole process. We have to keep changing it, so it gets very confusing. But when I say VX770, I mean Kalydeco. And you can see here lung function in patients taking variety of doses of VX-770. And the black line, it's probably very difficult for you guys in the back to see because these are small, but the black line you can see people who are on placebo, so a drug that didn't have, it's taken, has no effective components in it. And all these slides are saying the same thing, just in a different, analysis, that patients on VX-770, you can see there's a very significant increase in their lung function compared to those patients who are on placebo. And that effect is sustained at 14 days. And then in the slide on the right, you can see that even after 28 days, the effect is still being sustained. And so this is what is so amazing about Vertex 770. They've opened that gate and we're able to normalize some of the function of the CFTR protein. And so just in summary, ivacaftor or Kalydeco, it's a potentiator of CFTR, and it's shown an increase in lung function and a reduction of pulmonary exacerbations in patients on study drug. And the FDA approved this in January of 2012, and it's now clinically available for patients 6 years and older who have a G551D mutation. So some other exciting things that are happening in 2012 is this clinical and functional translation of CFTR project, and it's called CFTR2. And so this is a database that's created by the Cystic Fibrosis Foundation and investigators at Johns Hopkins, and it contains information on over 35,000 patients in the United States, Canada, and Europe. And it includes their clinical information, which we put together with their genetic information. And we're going to flip to the next slide, and we'll see why this is useful. So when we have a database that contains so much information, we're able to, you can see here, find new mutations in CF that may be disease-causing. It also allows us to be able to pair clinical symptoms with certain gene mutations. So we have more information about each individual mutation, and we'll be able to talk to patients in more detail about what having a certain mutation might mean. And so if you'll flip to the next slide, I'll show you what the website looks like. And so this is going to be very difficult for you guys in the back, but I just wanted to give you a sense of the information that comes up. And this is available for patients, for providers. You can go in and search the mutation database. So if you know your mutation, you can put it in. And what comes up, you won't be able to read this back there, but it tells you how many patients have the mutation in the database. What this says, I put in G551D because that's, a mutation of interest lately. It shows that Kalydeco has been approved, and it goes over specific mutation combinations. If you have one G551D and then a delta F508 with it, it'll go over some of the information of what clinical symptoms will be like in these patients. And so if you click, this is the general view, and if you click over one more, this is the scientific or medical view that we have here. And so clinicians can use this to figure out clinical characteristics, mutation characteristics, and this just gives a vast array of information for both patients and providers when you want to research CF mutations and what we can expect. And as this database grows, right now it only contains 160 of the most common CF mutations, but what we're hoping is that more information will get added and we'll be able to have information on all the mutations that cause CF, and we'll be able to use this as a tool in the future. So we've heard about what exciting things have been going on across the country, across the world. Now let's talk about what's been happening at Maine Medical Center. And some of you are very familiar with what's been going on because you've been involved in these projects. One exciting thing that we're doing right now is an investigator-initiated study written by our very own Dr. Zuckerman. And we're looking at infection control in CF outpatient clinics. And so this is going to be a very important study in making recommendations about infection control in our clinical practices. And so I see a lot of people who have participated already in this study, and if you haven't, we are still enrolling for this study, and we would love to talk to you about being a part of it. And so we're also testing a new anti-infective against Pseudomonas, and that is inhaled levofloxacin. So we have 2 studies going on right now that we just finished enrollment on, and so we're in the study phase, and we are looking at if inhaled levofloxacin is better than placebo in treating a Pseudomonas infection. And then the second study is looking at it against Toby to see if it's any different in the outcomes versus inhaled Toby. Now the eye care study is looking at adherence, and as Karen had told us, adherence is incredibly important in the CF population, and this study focuses on adolescents and their adherence to treatment and medication schedules. And so what this study does is it puts the adolescent in control of what they want to work on. And so we're sort of empowering them to think about what areas could they be better at in their treatments, and let them take charge of, you know, what they want to work on. And so this study has been very eye-opening for us because it has taught us what things we need to teach better, and we hope that it's also helped our teens figure out how to be more adherent to their treatments and enjoy the process. So the next study is another investigator-initiated study that we're teaming up with on Dartmouth, and we are looking at iron supplementation in cystic fibrosis. And that one is the adult team has joined forces with Dartmouth, and we're exploring whether taking an iron tablet by mouth for 6 weeks improves the amount of iron in the blood cells in the bloodstream, and if lung function and the amount of iron in the mucus you cough up changes when taking an iron supplement. And another investigator-initiated study that we've been working on that Dr. Zuckerman also wrote was looking at the complications associated with PICC lines and implanted vascular access devices. So that is a retrospective chart review looking at what complications come up with those procedures. And Karen had just given you a very good overview of inhaled mannitol, which we hope will be coming down the line and getting approval from the FDA very soon for use. And so what's in the works, what's coming up for you to be involved in? And you can see this is a slide from the TDN, who is the Therapeutics Development Network, which is a network that provides funding to CF clinical centers in order to do research projects. And so we are a TDN center here at Maine Medical Center. And you can see what's coming down the line. Vertex has a lot of studies going on looking at the ivacaftor we have it coming down for 2 to 5-year-olds with G551D, and so I think they're making a liquid formulation of that for G551D, and then they're going to also test it in other mutations, some of the other gating mutations, to see if it works the same as it would in the G551D. So it's very important, like we said, to know your mutation, because we have a lot of studies coming down the line that are going to be mutation dependent. And so Vertex 7070 has a bunch going on. There's an enzyme study that's going on that we're not going to do here at Maine Medical Center, but other sites in the country are going to be working on that. And then Novartis, who are the makers of Toby, have a mystery agent coming down the line, which is going to be an anti-infective treatment for Pseudomonas. And so that hasn't been revealed what the mystery agent is yet, but we're hoping to find out soon. And then Novartis also has an inhaled powdered Toby. So you heard about inhaled powdered mannitol and what the advantages of a powdered drug is. And so they're thinking of developing an inhaled powdered Toby, and other countries actually already are using inhaled powdered Toby. And so that will be an exciting study that we're hoping to get here at Maine Medical Center as well. And so now that you know about what research is coming up, let's talk about why it's important that you participate. So for all the drugs that you or your children are on, Toby, Cayston, everything has gone through this process coming through the drug pipeline, and years have been spent on developing these drugs. But if you guys don't participate in these studies, we have no way of getting the drug from point A to the point where we can prescribe it clinically. And so it's very important to have patients involved in clinical research trials so that we can get these medications through this process quickly and get them to you clinically. Because you, can you hit the button? Are the key to success. And so thank you all very much for your attention today. Does anybody have any questions?
So if you would like, or would you like to participate in a trial, or you'd like your trial to, how do you go about doing it?
So, yeah. So she wanted to know if you want to participate in a clinical trial, how do you go about doing that? So I know both Karen and I, we screen all the patients in our clinics when we have a new trial and we approach them. But if you are particularly interested in research, you can contact any person on the research team or on your clinical team, your physicians, the nurses. They're all going to know how to get in touch with somebody in the research realm. And so basically, talk to us. We will tell you what we have. If you're looking for something, you know, at another center, if we don't have it, we're able to get you access to that as well. And so, you know, just talk to your physician, talk to your research coordinator, talk to your nurses, and they'll be able to get you the information you need or direct you to a resource for that. Anything else? Yes.
Could you please repeat the website address for that
CFTR2? Yeah. Do you want to back it up? Because I don't know it off the top of my head. Back it up. There. So right here at the bottom, it's http://cftr2.org. And you could probably also Google CFTR2, and it will pop up there. Mm-hmm. Yes.
I have a question about the mannitol.
Ooh, good.
And this may be jumping the gun, but is that something, it sounds like it does a lot of the same thing as the hypertonic saline does. Is that something that you would take hypertonic saline and mannitol, or do you pick one or the other in terms of convenience?
During the study, patients could not take hypertonic saline while they were on mannitol. How that will work clinically, maybe Dr. Zuckerman would be the best person to answer that question once it's FDA approved.
Yeah, so right now, they're not to be used together because they haven't studied together, but you're right, the mechanism is similar. The hope is that the dry powder mannitol would have a more durable effect because it isn't absorbed through the airway as quickly as hypertonic saline.
Yes.
Someone have an estimate when that powder would be coming online? Is that too hard?
At this point, it's at the FDA, correct, Dr. Zuckerman? We're hoping this year, but do you have any more information?
Right now, I haven't heard any more information, but the trouble with most of these trials is that the numbers are quite small. So they're looking for follow-on study results and things like that. So we're hopeful.
We're hoping.
The Caiston story is kind of illustrative of how long it could take after we have the data to getting it actually approved.
Any other questions? No? Thank you very
Thank you.
much.